Hello, everyone, and welcome to our 6 o'clock presentation. Hello. Welcome to our 6 o'clock presentation with Dr. Matthew Harms. He's going to be speaking to us about 30 years of genetics in ALS. What have we learned and where are we going? I will be your moderator this evening. My name is Debbie Schlossberg, and I'm the Chief Care Services Officer here at the ALS Association Greater New York chapter. Dr. Matthew Harms is an associate professor of neurology at Columbia University Medical Center. Many of you undoubtedly know Dr. Harms from your visits to the ALS clinic at Columbia. But Dr. Harms also has a very long history in the field of neurogenetics. And as a researcher, he's going to be speaking with us tonight about the field of genetics. We're always very, very grateful to Dr. Harms for his time and attention to the entire ALS community. We'll be handing it over to him in just a minute, but I want to remind you that at the conclusion of this presentation, we'll be having a question and answer session. So any questions that you have, you can please enter using the Q&A function at the bottom of your screens. Thank you, Dr. Harms. We look forward to learning from you.
Thank you, Debbie. It's always a privilege to come and speak at these educational symposia that you put on. The entire community is very grateful, and I hope that by the end of today, I'll have elucidated some of the complexities of genetics in ALS. Can everyone see my main presentation screen? Great. Well, for those of you who looked at the Whova app or the agenda that was sent out, you will have noticed that my talk title has changed. It used to be 30 years of ALS genetics, separating the wheat from the chaff, and was going to focus on how we are learning some lessons about genetics and having to unlearn some things that we have thought we had nailed down from the past. But as many of you know, we've had exciting news in the era of genetics with the approval of tofersen. And so instead of giving that particular talk, I wanted to shift the focus just a bit and answer the question, what have we learned and where are we headed? Before doing that, though, I do want to just give you some relevant disclosures. The first of which is that my research laboratory has been funded by several companies, including Biogen and Ionis, as well as many foundations, including the ALS Association, which really gave me my start in ALS genetics, along with some of the others that are listed here. And then I have, like many experts in the ALS field, provided input and guidance as a consultant for many of the clinical trials that some of you have participated in, and will point out if none of these I think are going to influence how I talk today, but just so you know my background. So it's been 30 years since the first ALS gene was described, almost to the month. And so I wanted to just begin by discussing the historical timeline of human genetics and ALS and how they've really interdigitated over time. So going back to 1865, as many of you probably learned in high school biology, Gregor Mendel described the principles of hereditary using all of those fancy P cross experiments that we all memorized back in the day. But it really wasn't for another 40 years or so that we really started to understand that chromosomes within the nuclei of our cells were the basis of that heredity. It was another 50 years before we understood how the chromosomes were composed of DNA and that DNA was part of a double helix. And it wasn't until another 30 years that we were able to map the first human disease gene. That was for Huntington's disease. And I'll just point out that even though it was discovered all the way back in 1983, we still as yet do not have a genetic therapy that works for that particular gene. And then fast forward to the early 2000s, and we first started to see whole genome sequencing, and now that's almost a standard component of what we do in patient care. Now on the bottom, I'll illustrate how the development of ALS has taken place. Many of you may not be aware that it was all the way back in 1869 when a French neurologist named Jean-Marie Charcot first described what we now call ALS. And it was quite some time before that was on the radar screen of many people, really until Lou Gehrig passed away from the disease in the 1940s. I just put the founding of the ALS Association on here because they've played a very instrumental role in the research and development that's gone on since their founding. And then point out that in 1993 is when ALS genetics really got off to a great start with the discovery of the SOD1 gene. And now, almost exactly 30 years to the week of its discovery, we now have an FDA-approved medication for that particular form of ALS. So probably if you've been coming to these over the years, you'll remember my ALS gene discovery timeline. I've altered it a little bit for this year. So here back in 1993, when I was graduating from high school, the SOD1 gene was discovered and reported in this pair of papers. It's always fun to kind of go back and see what the fonts looked like back in the day, and you can fortunately do that. Dr. Brown's group and Tipo Siddique's group discovered that quite a few families were caused by mutations in the superoxide dismutase 1 gene. At the time that this was discovered, this graph here illustrates the number of ALS papers in the scientific literature by year. And you can appreciate that before the discovery of SOD1, things were kind of flat. And on average, there were somewhere between 100 and 250 papers devoted to ALS each year. With the discovery of SOD1, things really began to take off. And you can see just over time, the amazing steep development of ALS literature. You'll also reflect in this curve of increasing papers and interest in ALS, the increasing discovery of ALS genes. In fact, they almost overlap, and I probably should at the next iteration merge these over one another. And you can really appreciate that it's the genetic discoveries in ALS that have drawn people to the field for research and have really spurred many of the increases in knowledge that underlie the field today. I think you can appreciate that cruising along here, there were some increases, but with SOD1, there was a steep increase. You'll probably see that right around the time that TDP43 was discovered as an ALS gene, there was another big increase in the number of papers coming out per year. And same with the C9orf72 gene. It's almost as if every single time we discover a major ALS gene, more people get interested, more research gets funded, and more things get accomplished in ALS research. Now with some 35 or 40 genes that have been reported to cause ALS, we're also explaining an increased number of people with the disease. When I first started in ALS genetics, we were cash strapped. Actually, the sequencing technologies were extremely expensive, so we could only use them in rare cases. And so back when I first started doing ALS genetics, we studied 42 families and 350 people who did not have a family history of ALS. And we found causative gene mutations in just over half of the families, and in about 10% of those individuals who had no family history in what we called sporadic ALS. That was all the way back in 2015. Fast forward now to 2022 or so, when I carried out another analysis, this time not of 500 people, but of 5,500 people with ALS. And interestingly, these proportions are basically unchanged. They look almost identical. And that's true with one exception, and that is that this largely reflects the European ancestry, predominantly Caucasian population that I was studying when I was at Washington University in St. Louis. And now with these 5,500 ALS individuals, an increasing proportion of them come from non-European populations, which is hugely important. And what I can tell you from analyzing people who are not of European descent is that C9orf72 is somewhat rarer in those other groups, and that SOD1, TDP43, and the FUS gene are more common in non-white populations. So we do have to take into consideration people's what we call geo-ancestry, or race and ethnicity, depending on how you think about it, into consideration in terms of how likely it is that the genes we know about today will be found on genetic sequencing or testing. These, are the main ALS genes shown here, but increasingly the ones we're finding are rarer. But because we're finding these genes, we also now have the FDA-approved tofersen for the SOD1 gene, shown here, which is the second most common cause. There are clinical trials actually happening for C9orf72 FUS, and then another gene that it's not listed or represented here called ataxin-2. And excitingly, in a program called Silence ALS, which is run from here at Columbia University by my colleague Neil Schneider, there's early development targeting the TDP443 gene, also known as TARDBP, the Annexa 11 gene, the KIF5A gene, and several others. And I would point out that some of the other rarer genes that aren't listed here also have early development projects at other institutions as well. Most of these development projects are focused on using a technology called antisense oligonucleotides, the same type of molecule that was just approved as tofersen. So I thought it would be useful to grab the very lovely diagram that the ALS Association website has on antisense oligonucleotide therapy to illustrate how it works, just so that everybody has it clear in their minds. So shown here on the left-hand column is the situation where there is no mutation in the DNA, so the normal circumstance. The DNA has no mutations, and when the body needs to use that DNA to make a normal protein, it first manufactures a copy called mRNA, and then that mRNA becomes the template from which the normal protein is manufactured. Now, when there is an ALS-linked gene mutation, shown here in red is the abnormal DNA, it leads to a mutated copy of the RNA, and then that leads to a bad template for making the protein. And the protein oftentimes becomes misfolded. It kind of crunches up and becomes toxic to the cell in some way because it's misfolded. Now, an antisense oligonucleotide can be designed to recognize that mutated RNA. And when you infuse it into the spinal canal, it gets into the neurons. It will then stick to the mutated RNA and block the body's ability to use it as a template. And so you won't manufacture the abnormal toxic protein, and instead we'll just have a little bit of the normal protein coming from the good copy of the gene. And hopefully that will be enough to keep neurons healthy. So it's this type of therapy that's driving the field. There are others. There are gene replacement strategies for other ALS genes. In this circumstance, the problem is a toxic protein. For other ALS genes, the problem is too little protein. And so in that sort of situation, you can't use the antisense oligonucleotide to block the production of protein. You actually need to boost the production of protein. And so there are other strategies, including virally delivered copies of the gene, that are being explored for some of those other types of ALS. So with one FDA-approved therapy already for the SOD1 gene, several additional ones with clinical trials ongoing, and lots of development for the rarer types, I think it's a good time for us to pause and discuss genetic testing. Now, if you live in New York City, you're likely to have already undergone genetic testing. We tend to be pretty advanced, and the last analysis of the Columbia Clinic showed that 93% of people coming to our clinic have undergone comprehensive ALS gene sequencing. That may not be true at every institution. And so the ALS Association, recognizing that, funded myself and some collaborators, Jennifer Rogenbuck, Frida Eubank, and Stephen Kolb, to write the very first ALS genetic testing guidelines for North America. So this began with a systematic review of the literature. I showed you the climbing number of ALS papers. We used some algorithms to take that number of 33,000 and find the 10,000 that were most likely to be relevant to this process. And then, believe it or not, we read the title and abstract of all 9,000 of those. It was extraordinarily time consuming. It took us more than a year to accomplish that. From that, we were able to pull close to 1,800 that warranted reading the papers. And basically from there on, we culled it down to around 263 that had all of the most important information to guide these testing guidelines. And I won't go into them in detail. Those are for neurologists. Our goal is to get them to do more genetic testing using these guidelines, but they addressed some very important questions. First of all, what should genetic counseling include at various stages of testing? These are some very important questions. How much do people want to know? What do their families want to know? What are the implications for their children, for their other relatives, for their insurance? And so we will be providing very clear guidance to neurologists and genetic counselors and others working in this field as to what should be covered before testing is offered, And then what should be covered once testing has been completed to make sure that people living with ALS and their families understand exactly what was found, what wasn't found, the limits of the testing capabilities, and directions for the future. The guidelines also answer who should be offered testing and what should testing include. And we've made the very strong recommendation that now with an FDA-approved therapy and these other clinical trials coming along, that all people living with ALS should be offered testing, that they should be tested with a panel instead of one gene at a time, and that these 4 genes should be included in every circumstance, but that additional ones are also likely to be helpful. So here in the United States, there are multiple testing options. We actually lead the world in these testing options. There are 3 commonly used commercial laboratories, though there are others, and 2 of these offer free testing if criteria is met. And so if you have not yet been genetically tested, haven't had that conversation with your neurologist, definitely bring it up, because at the moment, 2 of these are free. It's relatively straightforward, and with the appropriate counseling, they can walk you through whether or not testing makes sense in your circumstance. Most of these commercial laboratories test an entire panel of genes that have been reported to cause ALS, somewhere between 24 and 31. I don't think it really matters. I don't want anybody to be worried about which lab does their testing, because by and large, the core genes, the ones that we are certain cause ALS, are all represented on all of the panels. Where they differ is how liberal they've been in terms of pulling in other gene mutation or other genes where the evidence is not quite as strong that they are causative of ALS. So what we would call some candidate ALS genes. So if you undergo testing at a US testing laboratory, what are we likely to find? Well, about 5 to 10% of the time, we get a positive result back. That depends again on your ancestry, as I was alluding to earlier. And an equal number of people will receive a result back that's called a variant of uncertain significance. A variant just refers to a place in the DNA where there's an abnormality that's different from the majority of people in the world. And it's of uncertain significance because we don't know whether it causes ALS. It might, or it might not, depending on some of its characteristics and what we learn over the course of the next several years as we research variants of uncertain significance intensively, trying to turn variants of uncertain significance into understandable and interpretable gene mutations. We would love for there to be no variants of uncertain significance. So we would either be able to look at the mutation and say, yes, this is associated with disease, or no, it's not. There is a time period right now when many of thousands of people around the world are getting their genome sequenced, and we're learning about all of the diversity in people's genomes, where we won't be able to always provide certainty. And certainly clinical laboratories and neurologists that are working at ALS centers that don't specialize or focus on ALS genetics may not have all of the information that they need to help patients understand if a variant of uncertain significance is identified. So my colleagues here at Columbia and myself have created something called the VUS-SOS, Variant of Uncertain Significance Second Opinion Service, so that any of our colleagues anywhere in the United States who have a patient living with ALS who has a variant of uncertain significance that they would like help interpreting to see if there's additional information about it, and be able to give that back to patients to understand more easily. All they have to do is email me, and I'd be happy to take a second look at it to provide that second opinion about whether we can interpret it with more clarity than what the laboratory where the testing was done could provide. Genetic testing also gives me the opportunity to talk about people who may find out that they have a gene mutation or are at risk for carrying a gene mutation but don't have any symptoms. And to introduce the concept of asymptomatic or pre-symptomatic ALS gene carrier studies. So here at Columbia, we have a study called the ALS Families Project. And our goal is to identify mutation carriers, mainly the siblings, kids, and parents of people who have gene mutations in ALS, who are at risk, increased risk of developing ALS. They may be carrying that exact same mutation. We believe that if we study these individuals who carry the gene mutation but don't have any symptoms, we'll be able to discover the very earliest things that go wrong inside of neurons, the very earliest stages of ALS, with a goal to be able to intervene at that moment and to prevent the development of actual symptoms and to prevent ALS. In fact, the ALS Association has funded a project here at Columbia, as well as at Mass General and at Washington University, called Prevent ALS, which is a collaboration between our ALS Families Project and those other two sites to provide free and confidential testing of relatives of people who have gene mutations, so that we can identify mutation carriers and include them in studies and provide counseling about every aspect of the disease process. The goal is to collaborate with other similar studies to make sure that we have the best chance for discovering those very earliest steps of ALS. And if you're in a family that has a familial ALS, or you know of a relative that carries an ALS gene mutation and would be interested in connecting with us for that type of a study, you can email alsresearch@cumc.columbia.edu, and we'd be happy to talk about whether this would make sense for you and your circumstances. So we've talked about the history of ALS gene discovery, and I want to just ask one basic question that occasionally people ask me is, if we already know so many ALS genes, why do we need to keep studying it? Why do we need to focus on this? And I'll just show this diagram. In the center is actually what a motor neuron looks like. The yellow ball is the nucleus where all of the DNA is stored. The yellow area, oh, I have a pointer, sorry. The blue area here is called the cell body, and then this portion that runs all the way down here is called the axon. And if you can envision, basically this axon runs to the muscle, and when it fires, it tells the muscle to contract. And by identifying all of these ALS genes that I've been talking about, we've been able to put them into different categories based on their function. And when you start to assemble them based on their function, you start to see themes. We start to see that processing of RNA, that intermediate step between the DNA and the protein, is a major theme in the function of these proteins. Protein homeostasis, basically how the cell deals with the toxic proteins that I showed you. The ability of the cell to ship things that are made up here in the cell body all the way out to the muscle end of the axon involves something called axonal transport, basically a railroad track that runs down the axon to deliver the goods to the very end of the neuron. And a similar process called endosomal trafficking, which ships things forward and back along that track. So by studying the ALS genetics, we've actually been able to zero in on these 4 or 5 mechanisms as of major importance, not just in genetic ALS, but in many, or if not all, ALS cases. And I would point out that by studying these genetic forms, we've also, not necessarily directly by lining up the different gene names, we've also been able to implicate lots of other functions within the cell that are going wrong in the majority of ALS patients. Some of these are illustrated here in another neuronal picture. And I would just say that every single one of these boxes that you see here has at least one, if not a dozen companies working on drugs that modulate those processes to try to turn back the clock or to prevent the development of ALS. So by studying the genes and the genetic forms, we have zeroed in on these very highly important pathways, and those pathways are now the target of drug development like we've never seen before. Now let me return to the ALS gene discovery timeline to talk a little bit about the multiple phases and what we've learned in those. So here's that same curve that I've showed since SOD1 was discovered. And I think you can appreciate that there's a change in slope. Slope just means the slant of the line. And so here's one slope, and here's another slope. You can kind of see these different inflection points. Phase 1 of ALS gene discovery was driven by very large families, something called linkage analysis that requires those large families, and then painstakingly slow gene sequencing. In fact, one individual postdoctoral fellowship, which was generally 2 or 3 years long, would do part of each of these steps, and then it might take a couple of them in order to get through the sequencing. And that's why, one of the reasons why, the discovery of ALS genes was relatively slow in that first phase. By the year 2000, we had the human genome published, and that helped the painstaking sequencing to get a little faster. But it really wasn't until stage 2 that the pace of gene discovery really took off. By this time, we only had small families, so we couldn't do that linkage analysis. Instead, what we had to do was look across multiple families for shared genetic problems. But by then, sequencing had become much easier. The genome was by and large hammered out. And so you can appreciate that gene sequencing and the discovery of ALS genes really took off at that point. But we're not in phase 2 anymore. We're by and large out of small families. Sequencing, however, has become easier. And so now we're in phase 3, where we're studying people living with ALS who have so-called sporadic disease, no family history at all. We're able to do very quick and relatively cheap, complete, comprehensive genetic sequencing. And then we're relying on statistical power in order to identify new ALS genes. And I just want to talk a little bit about how we do that so that you can appreciate basically almost every gene that's shown here was discovered by this method. And so you can understand the pitfalls and limitations of that. I'll just walk you through essentially how it works. And I promise you, you're going to be able to follow it. Sometimes people get nervous or panic that they're not going to be able to understand kind of these statistical models. They hear statistics and their brain starts to shut down. But I promise you that you'll be able to hang and kind of get the general gist of how we discover new ALS genes. So basically, when we do whole genome sequencing in thousands of controls, I've only shown a handful here. Each one of these lines represents a different individual who does not have ALS. And thousands of people who do have ALS, we always find genetic differences. And so here, almost every single one of these individuals in this blue gene that we're looking at has a genetic difference from what we expected. I've shown all of these as yellow stars. And I'll bet if you kind of squint and appreciate, it looks like the number of abnormalities in the controls is quite similar to the number in cases. And that's what we expect to see. But we know that all differences in the genome are not equal. Some of them do absolutely nothing to the gene. We call those benign or harmless. And then some of them are going to be absolutely lethal to that protein. They're going to be such a bad mutation that it makes it so the protein doesn't work at all, or that it acquires one of those toxic functions like I showed earlier. And so if we have a definition of what a harmless mutation looks like and what a damaging mutation might look like, we can go back and reassign those findings in the cases and the controls and then do a comparison. So if basically we make the harmless variants a green star to illustrate that, and the red ones damaging, and then we reassign them across these thousands and thousands of patients, now you can start to appreciate that in the control group, there are only a handful, only 2 of the damaging abnormalities amongst all of those people. Whereas on the cases side, the ALS side, there are 7 or 8. And if you do statistics across the whole genome and find the regions of the genome where those abnormalities really differ between the cases and the controls, you've zeroed in on a new ALS gene. Now, it doesn't tell you which mutations are the problem, but it does tell you this gene is important in ALS, and we can do many of those same things. What is its function in the cell? Is it a good target for developing drugs, and so forth? So we, in 2013 through 2015, decided to apply this very new principle. And we carried out the ALS exome consortium sequencing. There were 9,000 participants in that study. Only about 3,000 of those were people who had had ALS. For the first time in ALS, we were able to assemble lots and lots of DNA from many different centers and kind of began the era of collaboration that continues to this day. I won't go into this in much detail, but basically this red line is where we expect all of the black dots to end up on if there's absolutely no difference between people with ALS and without ALS in terms of how many mutations that look bad are present. And you can appreciate that many of these genes start to rise up off of this red line. And the higher up they go, the more implicated they are in ALS. And I don't expect you to be able to read this very well, but you'll appreciate that the very top thing that we identified in this study was the SOD1 gene. So if we had not known about the 1993 discovery of SOD1, we would have discovered it by using this method. Even more exciting, we found the TDP43 gene. We also implicated two brand new genes, TBK1 and NEK1, which have gone on to become very important tools for understanding ALS. So the more people that are participating in these studies, the more genomes that we've had a chance to study, the more we discover. So in the next iteration, we took it from 9,000 participants up to 11,000 participants. And when we did that, we were able to, again, rediscover the SOD1 gene. But now we're starting to see some genes we haven't heard of before starting to come off of this red line, which makes them candidates for ALS genes. We didn't really expect to find definitive new ALS genes only going from 9,000 people to 11,000 people. And so now we're in the middle of the next iteration, which is 27,000 people. So almost triple the size. We've been able to do this because we've assembled all of the people sequenced here at Columbia, the original ALS sequencing consortium that was funded by Biogen, the GTAC study that the ALS Association funded for me to run out of Columbia, and then the New York Genome Center, where we used to have this annual get-together. And we're able to come up with 27,000 participants, about 6,000 of whom are people living or who had ALS. And what have we found? Well, excitingly, we're seeing many more genes rising up off of that line. This time it's blue instead of red, but same concept. The higher these little dots, the more significant the gene. And excitingly, 5 known ALS genes are now showing up in our statistics instead of just 2. And what that tells me is that as we increase the number of people and participants in these studies, we're discovering more and more ALS genes. And there are some extraordinarily interesting candidate genes that are starting to rise up as well, telling us we're heading in the right direction, that we need to keep going, increasing the number of people that have had whole genome sequencing. And importantly, increasing the number of non-European ancestry individuals, because we're learning a lot from individuals who aren't from Europe or have a European ancestry. So one of the things that all of this collaboration has done is led us to be as collaborative and as open source as possible, meaning we want people to have access to the data. And so those individuals who provided permission for us to show their genomes have been de-identified, meaning we've removed the ability for people to figure out who they were. And of those 9,000 ALS patients, 6,300 of them have given permission for us to have the genomic variants posted on atabdb.org. So any ALS researcher around the world can have access to this and look up and learn more about these ALS genes. So that's our current iteration. But we didn't discover new ALS genes with 27,000. And so we have in the works with other collaborators an iteration with more than 100,000 participants. So the folks that I already introduced you to who helped drive the 27,000, another group out of the NIH and UMass in Worcester, and then European collaborators at Project Mine are all working together to generate data in something called ALS Compute, which is already happening. The ALS Association provided funding to be able to aggregate all of our data in one location in order to make this possible. And I'm very excited that this is going to yield a handful of new ALS genes that will help refine our targets and help us get across the goal. So that's how we're approaching finding new genes that have a very large effect in ALS. If you have one of those mutations, it increases your chances of developing ALS 5, 10, 15, 20-fold. But it's also the case that there are genetic differences between ALS patients and people without that don't have as big of an influence. They maybe increase the risk of developing ALS by 1.5 times or 2 times. So it doesn't determine that you're going to get ALS, but it increases the risk. And there's also a lot to be learned from there. And I'd like to talk a little bit about that because it talks about and gives me an opportunity to illustrate how we're thinking about using genetic therapy to treat everyone with ALS, not just those people with rare gene mutations. So common variation studies are a lot easier to do in ALS. They've been, DNA has been collected now for decades. And so people have had kind of common differences in the genome looked at. And back when the first study was done with 13,000 individuals, we really identified, in this case, it's how tall this little peak is. This is called a Manhattan plot, so it's appropriate for the Greater New York chapter of the ALS Association. These look like skyscrapers, and these are the super talls down along Central Park. And you'll see that we only had a couple that got above this red line, which tells you how important they are. Now that red line in the latest iteration, which has 150,000 people, not doing whole genome sequencing, not looking for the rare variations, but looking at the common locations across the genome that differ. Now that red line basically has gotten all the way down here. So anything above this is a significantly risk factor for ALS. And you can see that now there are many more that are above that line and telling us which regions of the genome and again, emphasizing the pathways, which could be triggering ALS and could be great gene targets. You won't be able to appreciate it given how small these are, but over here in this little cluster here is a gene called UNC13A. And I want to use that as a jumping off point because UNC13A is a risk factor for ALS. People who have very common differences in the UNC13A gene, when they develop ALS, tend to progress faster than people who do not have those abnormalities. And we know that UNC13A helps the very end of the motor neuron live a healthier and more productive life. So it's all the way down there where the neuron is communicating to the muscle called the synapse. And it's been known for a number of years that if you carry these abnormalities, and they're not really abnormalities, they're present in 30% of people. So they're differences, let's say, but they're differences that cause problems. And if you carry those, your risk for ALS is increased, but we didn't understand exactly how that would happen. And now, just really over the last 6 months, that's become quite apparent. And it all comes back to an abnormal protein that we've known about in ALS for a very long time. If you're in my clinic, you've probably gotten tired of me talking about it. And that is a protein called TDP43. So this is, the red dots here represent the TDP43 protein. It's an RNA DNA binding protein. DNA lives in the nucleus, so not surprisingly, in the normal state, the TDP43 protein lives in the nucleus of the cell. So this is that cell body, and then this is the nucleus, and here are all the TDP43 molecules. And they're supposed to be there in the nucleus, with an occasional one will pop out into what we call the cytoplasm, this area of the cell, but then eventually it will go back into the nucleus, and another one will pop out and it will go back. And so there is some shuttling back and forth, but most of it is supposed to be in the nucleus. Now in ALS and in some forms of frontotemporal dementia, that has gotten messed up. And when we look at the cells under the microscope, we can see that there's very little TDP43 left in the nucleus, and it's clumped up out here in the cytoplasm, making big protein globs that are problematic. So if we could figure out how to push the TDP43 back into the nucleus, or keep it from clumping up out here in the cytoplasm, that might be one way to treat ALS. So people in the Gittler lab went looking to find out if there were any genes that controlled how easy it was for TDP43 to leave the nucleus and get caught out here in the cytoplasm. And one of the genes that they discovered was called ataxin-2, which is one of those little peaks that shows up on the Manhattan plot, the ones that tell us of a region that's a risk factor for ALS. If you have an intermediate repeat in the ataxin-2 gene, your risk of developing ALS is doubled, more or less. And so the Gittler lab asked the question, basically, if putting in ataxin-2 causes the TDP43 to leave the nucleus, what if we delete or reduce ataxin-2? And in fact, they were able to demonstrate that if you used an anti-ataxin-2 antisense oligonucleotide to decrease the level of ataxin-2, just like we're doing with tofersen for SOD1, that more of the TDP-43 stayed in the nucleus and less of it ended up in the cytoplasm, restoring some of the function of this abnormal process. Now, getting back to the other risk factor, the UNC13A that I mentioned earlier, we've also now basically figured out what's going on there, we think, and that is that when TDP-43 leaves the nucleus and goes into the cytoplasm, it stops doing a very important job, which leads to abnormal utilization of mRNA, and we call that missplicing. And when TDP-43 is out here instead of in the nucleus, several genes get misspliced. And genes that get misspliced have generally speaking, get misspliced in a way that deletes them. And so here are a couple. So when you have missplicing by TDP-43, a protein called Stathmin-2 gets reduced. That UNC13A protein that I mentioned gets reduced. And there are somewhere between 50 and 80 other ones that also get reduced. And each one of these processes theoretically could be reversed by an antisense oligonucleotide. So that even if we couldn't fix TDP-43's location, we could block the missplicing of the Stathmin-2, of the UNC13A, of these other proteins, and by blocking the missplicing, lead to their increase, which may repair how the cell is functioning, which may decrease the progression of ALS, or maybe one day even prevent it. People are working on this extraordinarily hard. Every ALS meeting I've been to this year has been largely focused on this topic. There are now clinical trials for the one circled in yellow for ataxin-2. It's the AL-SPIRE or ALS-SPIRE trial that you can look up. For Stathmin-2, the study is being conducted by Curalis and dosed its first patient just recently. We know of other groups that are developing antisense oligonucleotides to change the splicing of UNC13A and many of these other proteins, these others that are reduced as well. This process is occurring in 95% of ALS. It's occurring in the people with ALS who do not have an SOD1 mutation. So for those people with an SOD1 mutation, we have the possibility of treating with tofersen. And now the field is heavily focused on developing antisense oligonucleotides and other genetic therapy strategies to address everyone else, the 95 or 96% of people who don't have a mutation in the SOD1 gene. So hopefully over the course of my talk today, as I've talked about kind of the progression of the 30 years of ALS gene discoveries and where it's led us in terms of therapeutic development, you can appreciate that the last 3 decades have been instrumental. They have provided the why did I get ALS for many patients, not many patients, but many patients. It's helped us to identify key pathways that are central not just in genetic forms of ALS, but in all ALS. They've successfully brought therapy targets forward for rare genetic forms. And more importantly, I would say, they've opened up avenues for developing gene therapies that could address all forms of ALS. I've never been more excited about the prospects for genetic-based therapy for everyone with ALS, regardless of whether they have an identifiable gene mutation or not. With that, I would just like to say thank you to the ALS Association for funding my research over the years, as well as to these other organizations who have generously shared data or provided funding to generate the data and exciting discoveries, not just in my lab, but in others as well. And of course, because we rely so heavily on DNA, we are extraordinarily grateful to patients living with ALS who have kindly donated their time as well as their DNA to the types of studies we're doing. And with that, I will pause and answer your questions. Thank you very much.
Wow, it was exciting. It is exciting. It's a very, very exciting moment in our space. Can you hear me okay?
Yes, I can.
Okay. All right. We have a couple questions for you. And I want to remind our audience that it may not be too late. We may have a chance to get to yours if you want to enter any now. But let's get started. First of all, there was a question that relates to these pathways. And the question is, where do current medications, specifically riluzole and Radicava, not tofersen, what pathways do they address? What do they attack?
Yeah, so interestingly, riluzole and radicava have been, successful at slowing down ALS in some studies, despite the fact that they're not strongly implicated by any of the genetic pathways. So riluzole, we believe, works by decreasing glutamate toxicity mainly in the crosstalk between the upper motor neurons and the lower motor neurons. And radicava is suggested to work by eliminating free radicals and other kind of toxic molecules that are around the neurons. So they're not impinging on these specific pathways, at least that we know, but they are, helping out and generating a healthier environment for the neurons as they cope with dysfunction in some of the pathways that the genetics have implicated, right? So, as the cell is stressed out dealing with the clumping of toxic proteins, the radicava and riluzole mechanisms are kind of protecting them from other stressors so that they have a better chance of dealing with the genetic pathway stress. If that makes sense.
Makes sense to me. Okay. Next question is, what is the timeline for getting a new drug to be effective in our lifetime? $1 million question.
Yeah, right. I think the timelines are getting shorter. I think one of the things that's quite exciting about the FDA's approval of tofersen is that it was based on a surrogate marker. It was based on the fact that it lowered neurofilament, not that we could yet tell that it was helping with strength. And I think everyone from the FDA, even though I know there's lots of frustrations with the FDA, everyone from the regulatory community, the institutional review boards at universities, the drug developers, everyone's pushing that gas pedal as fast as possible. And so I'm not a drug developer. I help find the genes, help discuss kind of which ASOs might work best or what should be targeted and that sort of thing. But then, it's such an expensive process that it gets handed off to others. So I would leave the exact timeline to others. I think 2 years is the fastest that we've seen, and that's obviously shaved 5 to 10 years in some circumstances off the clock.
Okay, thank you for that. Okay, so here are a couple questions. I'll ask both of them at the same time. One of them is, what is the role of epigenetic factors? Another is, why do some viral infections trigger ALS?
Sure. Let me start with that one. So one of the things that I didn't talk about when my original talk title was separating the wheat from the chaff, one of the things that I was going to emphasize is that we've discovered implicated a lot of ALS genes, and as the genetic information has gotten better and more refined, we realize that they probably aren't associated with ALS. So one thing that I also want to emphasize is that genetic discoveries are not static. We're always trying to understand, were we right the first time or not? That's why we do validation studies, why we don't just do one study, declare it a truth, and stop. In these genetic studies that I was showing you, we always have something called a validation cohort. So you test it in one group of people, and whatever you find there, then you go and test it in a second, completely separate group of people to make sure that it's true. And when it comes to viral triggers for ALS, I think we're in between those phases. Some studies have suggested that yes, there are viral triggers. Other studies have said, it doesn't look like it's that strong of a risk factor. And I think we're still trying to validate and figure out kind of where it is in that process. Now, I'm more confident that epigenetic modifications play a role in ALS, and the main evidence for that is that the risk of developing the disease increases with age. And we know that the strongest influence on methylation and the other markers that contribute to the epigenome are age dependent. And so for sure, as people age and their methylation and their epigenome shifts, there's going to be a shift in gene usage, and that is going to facilitate the development of ALS. We don't have strong markers yet for which methylation changes are important. So we can't just go in and force methylation or force some other sort of epigenetic modification because we don't know which ones are important yet. But that's definitely one of the things on the planned agenda for the next iteration of genomic studies, is to integrate and to include epigenomic information and data.
So that sort of leads pretty much directly into two questions, which are questions of definition. One question is, can you define genomics? And the other is, what is precision medicine?
Yeah. So genomics is basically the study of anything in the genome. And so that's going to be the DNA. It's going to be how the DNA is coiled. It's going to be all of the proteins that are attached to the DNA and shaping how the cell uses the DNA. And, we have only had the structure of the genome, or the sequence of the genome, now for almost 25 years. And we're still unpacking little regions of it and trying to understand it. And so genomics is the study of how that relates to human disease and human evolution, kind of other aspects of DNA. And precision medicine is something that hopefully all doctors have been doing for all time. Precision medicine is the idea that you identify a subgroup of people who might have a better response to a drug, or who have a subtype of the disease that you can recognize, or who are less likely to have a particular side effect or more likely to have a side effect. And you target those individuals with the right drug. So you can think of it as the right drug for the right patient at the right time. And really, precision medicine, as a term, has gotten popular because now we have the ability to apply genomics into that. It used to be we would just apply what we knew about you from the clinic, like how fast you were progressing, or whether you had abnormal liver function tests, or different aspects that we could see just on a standard examination. Now we're hopeful that we will be able to apply genomics to find subgroups of people and predict who will respond or who won't respond to a particular thing, right? There's some indication, we studied lithium for ALS in 3 very large studies, when I was first getting started as a neurologist. And those studies were negative. But when people went back and looked, it looked like if you had a certain form of that UNC13A gene that I was talking about in my talk, there was a better chance that you would respond to lithium. And so maybe lithium is good for people who have that particular genetic difference. And so there's a second trial now taking place in Europe for people who have that genetic difference to see if, in fact, lithium helps those people. Mm-hmm. And so that would be a really early example of precision medicine for ALS, to be able to sequence your UNC13A gene, know whether you have that difference or not, how many copies of that difference you have. And then if you have 2 copies, if that study is positive, then we would say, well, lithium would be good for you, but not for you. And so we would be precise about who we give the medication to. Tofersen is a very extreme example of precision medicine, right? Because it's something that we find an SOD1 mutation, and then tofersen is applicable. So that's another example of precision medicine. It's just a rarer example. And of course, we want to move this to as many people as possible. So we're looking for things that aren't quite that rare.
So promising. Okay, so somebody was asking specifically about SOD as their treatment, and that is tofersen now. But somebody else asked a little bit more sort of logistically about tofersen, and they want to know if they can get it, if they qualify, what to expect, where could they get it, a little bit more of the down into the weeds of what the experience is.
And you're talking about with tofersen itself?
Yeah.
Yeah. So tofersen has been available now for all of 8 days from a specialty pharmacy. It was amazing, really. Basically, from the time that it was FDA approved to the time the pharmacy was shipping it was less than 5 days. So lots of advanced planning to make that possible. There are 5 or 6 sites around the United States that participated in the expanded access program. So between the end of the study and the FDA approval, people with an SOD1 gene mutation in some circumstances could get access to the medication for free. And so there were 5 or 6 centers, including Columbia, University of Pennsylvania, Emory, Mass General, there are a handful of others that already have experience getting this medication and dosing it for patients. But it's also the kind of medication that can be administered with the appropriate training nearby. And so there's on the Biogen website for tofersen, there's a clinic locator. And so clinics that have this up and running, who know how to give it, have posted themselves to that site so that you can look and see if it's how close it's available to you. Again, tofersen is utilized to treat people who have mutations with SOD1 only. It doesn't work for any other gene that we know of. There are some people who want to test it in everyone to see if maybe SOD1 plays a role in some of those others, but I think the data is probably a little too early on that front. But yes, it will be widely available, and actually, Biogen priced it about the same, just a little bit more expensive than Relyvrio and Radicava, kind of right in that same spot. So even though it's a gene therapy, it's not substantially more expensive than something that we're, drugs that we're using for all of ALS.
So we should also add to the questioner that if you feel that you are a candidate, that you should speak with your neurologist about it, and he or she can direct you.
Yeah, and I would say soon, right? We know that basically time is neurons. Like the ALS disease process continues, even when you're not necessarily losing strength, neurons are having trouble coping. And so you want to, basically, if you think you might be a candidate, definitely getting treated earlier is highly recommended.
Okay, now here's a question a little bit different. This person says, my sister was diagnosed with ALS this past year. She's 74 years old. There is no history in the family. Could you use my DNA in your research, even though at this time there are no signs of me having it?
The short answer is potentially. So essentially, if your sister underwent genetic testing and was found to have an ALS gene mutation, then not just your DNA, but your spinal fluid, blood, potentially skin fibroblasts, all sorts of things would be highly informative and very useful for our research. If your sister doesn't carry a gene mutation, then it could potentially be useful as a normal control, because you would obviously be very unlikely to be carrying a gene mutation. But if your sister has not had genetic testing, we wouldn't know how to use your DNA. And so having genetic testing having been performed in the person who has the disease is a major priority and kind of a first step towards us considering whether we can include someone who doesn't have disease, who's related to somebody with disease, in our research programs. And if you're interested, there are nationwide programs that are collecting DNA from anyone and everyone. So it's a program called All of Us. There are lots of ways to get your DNA into that. And they just released the first almost 500,000 whole genome sequences for Americans who allowed their people residing in the United States, who allowed their genomes to be sequenced and to be coupled with their medical record so that people could use these 500,000 Americans as controls for their studies.
Okay, I'm going to sneak in one more question, and the question is this: are there any mutations associated with specific ALS features like presentation, progression?
So there's one. So the D91A mutation in the SOD1 gene, if you have 2 copies, everybody gets slowly progressive ALS that starts in their feet, goes up the legs, and eventually includes bowel and bladder function. That's highly, highly, highly predictable. For all of the other gene mutations, they skew in one direction or the other. Maybe they're more likely to have predominantly lower motor neurons involved, are more likely to have predominantly upper motor neurons, or more likely to start with bulbar symptoms or upper extremity symptoms, but they don't have as hard and fast a rule as that one mutation. I guess maybe there's a second, and that's the kind of an early onset FUS mutation that can affect kids when they're teenagers and in their early 20s. That one is also relatively has some pretty consistent features. But one of the things that's challenging in ALS is that there's a lot of what we call heterogeneity. There's a lot of difference person to person, even within the same family with the same gene mutation, that limits, makes it the case that we need to study as many people as possible, really, to kind of understand what is causing that, what we call phenotype genotype correlation, what you see on the outside and how it relates to what the genes are doing, that study.
Fascinating. Well, I have to say, first of all, to all the questioners and attendees, if we didn't get to your question, we will definitely follow up with you. So no worries there. There's only so much time, and this is a hugely exciting topic. And that's sort of my takeaway too. In addition to all of the information and the education, it's the excitement and the promise that you've given us, Dr. Harms. This really, really is a moment in our space, and we're very lucky to have people like you who are on top of it all.
Well, thank you very much. It's a real pleasure to be able to join in today.
Thank you. Before we sign off, I want to remind everybody that in just a half hour, we'll be reconvening here for the screening of the PBS documentary, Matter of Mind, My ALS, and we will be joined at the conclusion of the film. Will be joined in conversation by one of the subjects in the film and her husband, Michelle and Kevin Stellato. So we hope that we will see you back here at 7:30. Again, Dr. Harms, thank you for everything, not just tonight, but for everything you do for our community.
My pleasure.
Good evening. Bye-bye.